Hi everybody. So it's a pleasure to have Madeline Balker with us today to give today's summer colloquium. So Madeline did her undergrad at the University of Waterloo, and she moved here to do her PhD in biophysics with Sid Goel. So today she's going to be telling us about extremely hot and exciting, biology concepts. So this CRISPR, which you might have heard about in the news. So she's going to tell us all about it and how it's a window into the microbial world.
Thanks, Kieran. Yeah, hi everyone, I'm Madeline. Is it going to be too bright in front actually?
Too bright.
Yeah, okay. I'm very excited to talk to you today about what I work on. So I'm doing biophysics, and so I'm going to talk a lot about biology, but this is relevant to my actual research, which we'll come to at the end. Okay, laser pointer is coming. So my group studies populations of cells, and I'm interested in bacteria and viruses and how they interact. And I'm going to talk about the biology of bacteria and viruses, and then that will lead us to talking about CRISPR as an immune system in bacteria. And also I'll touch on the genome editing part of CRISPR, which everyone is very excited about. And finally, I'll talk about some of the work that I'm doing myself. So this is a video of a white blood cell chasing a bacteria, a small black thing. And when we look around us, we see all this amazing life. We see life doing crazy things, but actually most life on Earth is invisible, and it's happening in a place that we can't see with our eyes. So this is just one example. Okay, the bacteria got eaten, so +1 for the human. This is just one example of the craziness that's going on all the time. Out of sight, even in our own bodies. And I'm not going to talk about the human cells so much, but we're going to be talking about bacteria. So I want to show you sort of some crazy things that bacteria are doing. So bacteria. Bacteria are small, single-celled organisms. Cool, thanks. And they are invisible to the naked eye. You look at them under the microscope. So maybe I can do this with my mouse. This is acceptable. Okay. So each of these are Streptococcus dermophilus bacteria. Each of these is about a micron wide. So this is quite small. Bacteria are the most abundant form of life on Earth. So it's estimated there's about 5 × 10 to the 30 bacteria on the planet. Second only to viruses, which we'll come to. And bacteria are extremely diverse. So this is a phylogenetic tree of life. And you might have 2 bacteria that are more different from each other than, say, you, an animal, and a mushroom, or you and an oak tree, or you and a slime mold, which is something else here. This one maybe. And archaea, when I'm talking about bacteria, I'm going to lump archaea in with this as well. Archaea are also single-celled microscopic organisms. And they are, on the surface, they appear similar to bacteria, and they were originally thought to be part of the same group, but then it was realized that they're actually evolutionarily distinct. So archaea traditionally are thought of as living in more extreme environments, but it's known now that they can live in lots of environments, including normal ones like our bodies. Okay, I think that's all I want to say about that. So the incredible diversity of bacterial and archaeal life means that they can also live in crazy environments. So there are species that can withstand extreme temperatures, extreme salt concentrations, extreme pressure, extreme radiation, all kinds of crazy things. So one example on the top left here is microbial mat at Yellowstone. So this is a hot spring in Yellowstone. This is obviously a crazy environment that you probably would not want to go in yourself, but there are lots of bacteria, which are the colored things here, and archaea that are happily living there. And same thing on the bottom here, this is an extremely salty environment, a saltwater evaporation pond. And the purple color is from an archaea species that uses a special rhodopsin light sensor protein to do a very efficient photosynthesis. Okay. Make sure I point it the right way. Cool. Yay. Okay. And these 2 are electron microscope images of bacteria, just to kind of give you an idea of the different shapes that bacteria can have. There's lots of crazy shapes as well. So one of my favorite examples of the the possible extreme tolerance of bacteria is a study that took a sample of the Mars rover surface, the Mars Curiosity rover, before it was launched. And they found about 10% of the bacteria they found there. They found like 300 species. 10% of them could withstand one or more of extreme UV, cold, or desiccation. So that's being dried out. So if there is life on Mars, it's possible that we brought it with us. And because of this diversity, bacteria can also get energy in lots of really creative ways. So people are interested in studying that as well from a technological perspective. So for example, for producing fuel or for processing waste. So the story of bacteria is going to get personal. You've probably heard that humans are about 70% water. Well, it's also, maybe you might not like to know this, but humans are actually mostly microbe. So microbial cells outnumber our own cells by about 10 to 1. And if you're wondering why we don't look like piles of slime, it's because bacteria are much smaller than our cells. So by weight, we're only about 1 to 3% bacteria. So it's still kind of a concerningly large amount, but it's okay because bacteria are actually good for us in our microbiome. So our microbiome, which is the collection of all the bacteria and archaea and microbes that live in and on our body, is really important for our health. So they do things like help us digest food, outcompete pathogenic bacteria, and they really are everywhere in our body. So this is from a big project called the Human Microbiome Project, where they took samples from about 300 people with different body sites and found basically lots of microbes everywhere. And in the context of health, it can also go wrong. So microbiome is becoming increasingly linked to a whole host of surprising health things, such as cancer, immune disorders, even mood things, gastrointestinal disorders. So the microbiome is becoming very important for health. But the kind of puzzling part is that microbiomes are very different between people. So two people that are both healthy can have a very different microbiome. It can depend on things like where you live, what you're eating, if you have a dog or not. If you have a dog, you probably share microbes with your dog. And even the same person over time can have a very different microbiome. So this image here is from a study where the two researchers sequenced their own fecal microbiome every day for a year, which is quite dedicated. And the colors on the top row here are just different groups of bacteria. So the first thing you notice is that there's differences between person A and person B. And also, if you look over time, which is going left to right, on the bottom here is kind of showing the changes. So blue is a decrease, red is an increase. It's a bit small, but this person was living abroad during this time, and they got basically a whole new group of bacteria that wasn't there before. And this person had a diarrheal illness, oh dear. And basically after that, it was short-lived, but after that, their microbiome was changed permanently. So even in the same person who's back to being healthy, your microbiome could look totally different. So one thing I want to talk about later is some of the things we're working on to use the interactions between bacteria and viruses to hopefully be a bit of a window into this puzzle. Okay, we talked about bacteria, we talked about archaea, some of the crazy diversity, but it's not just other bacteria that bacteria are dealing with in their environments. There's also viruses that infect bacteria. So this is an E. coli bacteria in green being affected by a virus. Viruses that infect bacteria are called phages, so I'm going to use phage and virus interchangeably. Yeah, so when I talk about how bacteria and viruses interact, this is what's going to lead us to CRISPR as an immune system. And just to note why people are interested in phages and their interaction with bacteria, indirectly, they're important because they affect the composition of microbiomes. So if a bunch of viruses are killing bacteria, this is going to affect the structure of a community. But also there are some applications. So one interesting area of research is called phage therapy, which is the idea that you can use viruses to treat bacterial infections in humans. And another avenue that's quite important is that bacteria are used in lots of industrial processes, and for example, like making yogurt. So to make yogurt, you have a huge vat of your favorite bacteria, and then if a phage comes along and kills your bacteria, that's not good. So there's interest in helping your good bacteria resist phages. So phages depend on bacteria for their survival. They reproduce by first attaching to a bacterial cell. And by the way, this is the same process by which viruses attack our cells. So everything on this slide is applicable to us as well. They inject their DNA into their host cell, and then the DNA contains instructions for the host to make more copies of the virus. So it copies the DNA, and then it makes more virus particles, puts them together, and then the cell bursts, and there are new viruses. So just as an aside though, this sounds pretty bad for the bacteria, but it's not always bad. Sometimes, instead of actually bursting at the end of this process, after the phage injects its genome, it'll integrate into the bacterial DNA and kind of just hang out there. So once it's integrated, the bacteria can divide and do its normal thing, and it doesn't get killed. And the virus is basically hiding in its genome as it is growing and dividing. And there are certain triggers that can cause it to come out, but it can also just live there for basically forever. And interestingly, there are several diseases, bacterial infections that are bad for humans, where the gene that actually makes the bacteria toxic is from a virus. So for example, diphtheria, cholera, botulism, salmonella, these all rely on a toxin that's actually coded for by a virus. So in this case, it's sort of like the virus is helping the bacteria, but I'm not going to talk too much about that. For the rest of this talk, bacteria are trying to get away from the viruses. So bacteria have evolved lots of ways to try to prevent themselves from being killed by viruses. So basically any step of this process, bacteria can get a mutation that prevents the virus from doing its thing. So they can mutate the receptor that the virus binds to. They can block the DNA from being injected. They can try and cut up the DNA by recognizing that it's different. There's lots of things that bacteria can do. And on the other side, phages can evolve away from this. So if a bacteria mutates its receptor, the phage can get another mutation that allows it to bind to a different receptor. And so you have this arms race kind of going forward and forward of bacteria and viruses trying to each outdo the other to survive. But what all of these things that I'm talking about have in common is that they basically rely on a lucky mutation. So either the bacteria is born with the right ability to defend, or it doesn't have it and it dies. But CRISPR, which we're now going to come to, is different in that it's an adaptive immune system. So I'll go through each step of this in a bit more detail, but in short, this sort of works like our adaptive immune system, where if you get chickenpox, you make an antibody that recognizes chickenpox, and then you don't get it again. But bacteria can do something similar, where they can take pieces of virus DNA and store them in their own DNA, so they have a record of bad viruses. And then the next time the virus comes around, they can recognize and destroy it. And something that will be important later, CRISPR is very common in bacteria. So out of all the published bacterial genomes, about half of them have CRISPR systems, or seem to have CRISPR. And out of all the published archaea, something like 85% of them have CRISPR. So this is an extremely widespread thing that bacteria and archaea are doing. Okay, let's talk about the steps. So the first thing that bacteria need to do is they need to acquire this piece of virus DNA. So this is where the terminology will come in. I'll explain about where the CRISPR name comes from. So the pieces of virus DNA are called spacers, and then the spacers are sandwiched between these pieces of bacterial DNA that are called repeats. And those are all the same, and they are partially palindromic, which is where the name clustered, regularly interspaced, short palindromic repeats comes from. And the reason why it's called that and not something cool like adaptive immune system is because this region was observed long before people knew what it did. So people said, oh, this is a strange looking part of bacteria. And then it was like 20 years later, about 10 years ago, that people actually realized that it was an immune system. So the bacteria gets pieces of virus DNA, puts them into this CRISPR locus, which is in its DNA as well. And then the next thing it does is it makes the sort of weaponized part of the system. So it copies this DNA into an RNA, and each RNA piece has that piece of virus DNA as well as that piece of bacterial DNA. And just as an aside, a DNA palindrome is basically because you have 4 letters and each of them are complementary to one other letter. So if you have, say, A's and T's and C's and G's opposite each other, they can bind to each other. So if you have a single DNA strand that's palindromic, it can basically fold over and make a loop like that. So that's the palindromic part of it. But These are assembled with Cas proteins, CRISPR-associated proteins, which the bacteria makes. And this is the part that actually floats around the cell and can actually carry the immune response. And that basically means that whenever virus DNA appears, this piece of DNA will bind to the virus DNA as it's trying to replicate itself. And this Cas gene cuts the virus DNA. So it's an enzyme that cuts DNA, and it cuts very specifically where it binds. So if a new virus appears that it doesn't have a spacer for, this will obviously not work for the bacteria. It'll have to acquire a spacer and then do this all over again. So it could be killed in the meantime. And the virus can also evolve away from this. So the virus can get mutations in the area that is being targeted by CRISPR, and then the CRISPR system will work as well. Okay, so before I go on to talk about some of the things that I'm working on, I want to talk about genome editing, because this is the part that made CRISPR famous. And if you've heard of CRISPR, it's likely in the context of editing. So this technology is very new. It's only about 5 years since it was even thought of. So the person who thought of it, Jennifer Doudna, was working on CRISPR as an immune system. So pieces of virus DNA go into the bacterial genome, are used against viruses. And she thought, what if instead of virus DNA, you replaced these spacers with some DNA that you've chosen that matches something else, like a bacteria or a human cell. Then you basically have something that can cut anywhere you want in anything you want. And that's, in a nutshell, the whole story of genome editing. But why is this impressive? You might think we've only cut something. How have we actually done any editing? In genome editing, it's really all about the cut, because you rely on the cell's ability to fix broken DNA to actually make a change. So for example, this is the process that would happen if you wanted to, say, insert a gene into a bacteria. So the first part, and what turns out to be the tricky part, is the cut. And that's the part that CRISPR helps with. And then you have to make a piece of DNA that has the piece that you want to insert and also matches part of the DNA that was already there. So these blue parts match, and this red part is the part that you're adding. And then the cell, when it's trying to fix this, will do something called homology-directed repair, which means that it recognizes this piece as being the same as this. So when it starts to fix it, it'll chew back a bit, look for the template, and then keep going. And then in the process, it will insert this piece. You can also do, say, a deletion. If you were to cut twice, then the cell will try to fix the two ends that are left, leaving out the part in the middle. So there's many more kind of tricky technical things that I'm not super familiar with that you can do with editing, but that's basically how it works. And also to understand why CRISPR is such a crazy thing for cutting, I just want to also mention how it was done before CRISPR. So people have been editing genomes for a while, but the way it was done in the past is basically you have proteins such as this one, which is a DNA binding domain coupled with a DNA cutting domain. Or this one, kind of same idea, DNA binding and DNA cutting. And this is a protein which you need to engineer specifically to bind specific DNA. So you can, in theory, cut anything you want. It's just that it's a much longer, more expensive, more complicated process. And so really the big change that CRISPR brought was that instead of having to engineer a protein, you can just change that guide DNA that it's using to match, and then you can cut anywhere you want. So that's CRISPR genome editing. Okay, and just one kind of application that I think is probably close for CRISPR editing. So there was a study where they took mice with sickle cell anemia. So sickle cell anemia is a blood disorder where you have some blood cells that are funky shaped and it's painful and debilitating. They took bone marrow from the mice, which is what makes blood. They edited the gene to fix the sickle cell mutation. So sickle cell anemia is a single mutation that causes it. And they injected that back into the mice, and now you have a population of stem cells that's making correct blood. And in something like sickle cell anemia, you don't need to fix everything. If you fix most things, or you fix more of it than used to be, you can actually alleviate the symptoms and cure the disease. So this is something that is, I think, close to being doable. Okay, so we've talked about bacteria and viruses. They're diverse, and there's CRISPR, which is to deploy that for the immune system. And now I want to come back to the title of my talk, which was CRISPR as a window into the microbial world. So now I'm going to talk about some of the work that I am doing. And some of the questions that we are sort of after are things like, how does CRISPR immunity affect populations of bacteria? So if you imagine some bacteria don't have it and some do, how does it affect things? Can we learn about these interacting populations by looking at CRISPR? And given that CRISPR is so diverse, so many bacteria have CRISPR, are all CRISPR systems immune systems? Can we learn about how it's being used kind of in the wide world of nature? And so the first thing I want to talk about is using CRISPR to map out interactions in microbial communities. And second, I'm going to talk about some work that we're doing to model. So for the first part, I was saying before that every microbiome is different. It's unclear what it means to have a healthy microbiome. And so what we're trying to do here is say that maybe the pattern of virus interactions can tell us something about the microbiome. And the reason that this is going to work is that you can think of this, if you just look at the DNA of a bacteria that has a CRISPR locus, you can think of it as an immunization record. So each of these pieces comes from a virus. In principle, you can connect those two viruses and say that this bacteria has been infected by which viruses. And something else I didn't mention that is also cool about this is that these spacers are typically always added at one end. So you also have a time dimension. In some sense, if you have lots of these things, you're looking back in time at the past history of the virus or the bacteria. So you could say things like, how is the virus mutating? Can you see if there are spacers that are kind of following the mutations of a virus? Things like that. So we did this, trying to match spacers to viruses with data from the Human Microbiome Project. Again, this is a big project where they sequenced about 300 people's 5 body sites microbiomes. So there are thousands of samples. And what I've done here, so what I'm showing is I've taken the data, so the DNA of bacteria in that sample, looked for CRISPR spacers, and then match those to a database of phages. And so each dot here is a match between a spacer and a phage in the database. And so this is just one sample out of thousands, but this is sort of the idea behind how this is going to work. And then we can say things like, what does this network look like? So what I've done here is rearrange the rows and columns to try to bunch them in clusters. So each cluster should have more things in it than outside of it. And here's the same thing from a different sample. So they are not maybe super different. And it's not exactly clear what we should say about this. We need to analyze much more data, and there's some technical things as well. But the idea, you could say maybe these networks are actually ecologically relevant. So maybe they actually correspond to niches of bacteria, where there's sort of like a subpopulation that's interacting with viruses, and you have another subpopulation that's not really overlapping with it. And perhaps there are differences between healthy and unhealthy microbiomes with this sort of metric. One reason to try it this way at all is that you could do this experiment in the lab where you take bacteria, you add phages, you see which ones can attack which ones. But there are some major problems with this. The first is that most bacteria are really hard to grow in the lab. So of the bacteria that we grow in the lab, there are way more that are living in lots of crazy environments, including our guts, that are very hard to grow. So that's one technical problem. Another problem is that those experiments take a very long time. So this is as simple as looking at DNA sequence data, which is easy to get these days. But one problem with this, as opposed to, say, those lab experiments, is that these databases of phages that I'm using might not be, so this is the options this way, might not be relevant to the environment, they might not be comprehensive, they might be biased. So the biggest improvement that we can do here is to actually get virus DNA, phage DNA from the same sample, from the same environment as the bacteria DNA that we're getting. So that's definitely something that we're looking to do. And a lot of this work is being done in a project called PhagePrexer, which is on GitHub. So if you're interested in coding, data analysis, you want to check it out. This is not really a plug, but it could be. Yeah, check it out. It's on GitHub. Okay. Now I want to switch gears again to talk about some of the modeling and simulations that we're doing. So this is still on the theme of using CRISPR to get an idea of how bacteria and viruses are interacting. And so this is, again, a diagram of how the CRISPR system works. And you can imagine that to bacteria, the only thing that matters is how successful they are against viruses, given that they have CRISPR. So what's the probability that a virus will still kill them if they have a CRISPR system? On the one hand, it matters to the bacteria a lot which spacer they have. So maybe there are some spacers that work really well, and some of them don't work as well. Maybe the virus has mutated some of them, and then they're no longer effective. So there's this aspect of the stochastic nature of individual bacteria, which we'll talk about at the end. But then there's also this sort of population level effect where you could think if the bacteria just boosts up the expression of these proteins, it can have more of these weapons, and then it can do better. So that's a population level effect that I'm going to talk about first. So with regards to expression of those Cas proteins, bacteria could just express everything all the time at very high amounts, and then they would have a super effective CRISPR system all the time. But you can also imagine that they might not want to do that. Perhaps there are environments where there aren't very many viruses, like maybe when the cells are not very dense, the viruses won't do as well, and it would be too energetically costly for them to express this all the time. So you might think that they want to increase their expression when, say, the cell density gets higher, and this is what has been seen in an experiment. The cells do actually boost the expression of their Cas proteins when the cell density is higher. Cas3 is one of these proteins. So we can imagine that maybe the expression of CRISPR systems has some dependence on cell density and ask what that could do to the system. And what I'm going to show is that you can add this density dependence and to our simple model and actually get an interesting result of bistability, which is not something we would have expected. Okay, so first, before I talk about CRISPR in the model, this is a simple version of the model with no CRISPR. And so we have three populations: bacteria, viruses, and nutrients, and it's in a chemostat. And a chemostat is a realistic situation because in some sense, nature is a chemostat. So in our gut, for example, you have nutrients coming in, you have things flowing out. So there is no real sort of fixed environment in nature. We can think of a chemostat as a good approximation to a natural environment. So again, the important thing for bacteria is how well the viruses do at attacking them, the probability of virus success. And that's this parameter PV. PV, the probability that viruses will successfully kill a bacteria. So I solved those equations at steady state. And I'm plotting it here. On the x-axis is this probability of virus success, PV. So for a certain value, for a low value of PV, viruses go extinct. Basically, viruses are not doing well enough to overcome the outflow of the system, and they just go extinct. But as you start increasing PV, the viruses start to do better, and the bacteria start to do worse. But interestingly, if you keep going, if you keep increasing the virus, like how successful the viruses are, the viruses go back down. And this is kind of surprising. You might not think like, oh, I make the viruses better, and why are they going down? But it has a simple explanation. It's because the bacteria are also going down. So the food that the viruses are depending on is going down as well. So yeah, this interesting thing is sort of one illustration of why modeling is necessary to show us things that we might not expect. Okay, so now to add CRISPR, instead of having one group of bacteria, there are now two groups, one that has spacers, CRISPR spacers, and one that doesn't. And so all the bacteria are able to get CRISPR systems, but only some of them at any time will have it. And now the probability that viruses will succeed is decreased if bacteria have CRISPR. So those are the two big changes here. And so if I do the same plot, so the solid line is what I showed before. The dotted line is now that I've added CRISPR. Bacteria are doing better, and viruses are initially doing worse. And then they're doing better out here because the bacteria are also doing better. So it's the same thing as before. They need bacteria to eat. So you could think of this already as talking about expression. So this axis is virus success probability. You can think of that as being due to increased or decreased CRISPR expression. So in some sense, going this way, think of it as increasing Cas expression. And you can already see the bacteria are doing better when the Cas expression increases. But now I'm going to ask and talk about what if we make that relationship stronger. So this red line, think of it as the same as the bacteria in the previous slide. So I've just flipped the axes and flipped things around. So instead now we have bacteria size on this axis and Cas expression on this axis. And again, as the virus success probability goes down or the expression goes up, bacteria go up. And so this dotted line is going to be the actual form of the variable CRISPR expression or Cas expression. And so in the simple case, if we just say it's constant, there will be one value of bacteria size that we get. And so on the bottom here, this is a numerical solution to the time-dependent equations. And so things oscillate and then they come to a single steady state. So now we could change the shape of this green line, which is the actual dependence of expression on density. And if we make it so that it increases a bit, we might still only get one fixed point. So here, they intersect here, so there's a single fixed point for bacteria. Sorry, these lines are different bacterial populations for different initial conditions. So we're just looking at bacteria now. It might come to a different fixed point, but it's still only one. But if we make a very strong dependence of this parameter on the bacterial density, then we can actually get two stable steady states. And this is a qualitatively different situation than before. And this suggests that there might be a way to experimentally test this if we look for parameters that control this switch here. So in this case, the only parameter that I'm changing is the steepness of the sigmoidal function, but it's not, there's probably not an easy experimental parameter that corresponds to that. So we have to dig a little bit deeper and ask what parameters might control this that we can actually test experimentally. And one reason why bistability might matter is in a situation where you actually want to, say, decrease the expression of CRISPR system. So for example, a biofilm is a community of bacteria that's very dense and it has a lot of health problems. So for example, cystic fibrosis, one of the problems is you get these persistent bacterial infections that are protected by a biofilm. And so if you wanted to, say, try phage therapy on a biofilm, because it's very dense, the bacteria might be expressing their CRISPR systems at a very high rate, and so you might want to try to bring that expression down before the phages can have effect. But if it's in this sort of bistable parameter regime, you might have to bring it down past where it's going to go back to the high expression state. So you need to get past this memory of the system that it was in a high expression state so that it has low expression. And then you can, in theory, treat it. Okay, so now I'm going to come back to this question of bacteria individually carrying which CRISPR spacer they have. So in experiments, you can sequence all the spacers that bacteria have in a population. So this is a single population of bacteria interacting with viruses, and the colors are different spacers. And so the important thing to see here is that over time, these are days in the experiment, individual spacers are going up and down. You can think of the colors as corresponding to spacers like this. And it's not clear if we want to model this, what we should do, because there's not really a steady state that we can pull out. There's sort of this unpredictable turnover. But if we forget about which spacer is which, and we just look at the distribution of sizes, so how many spacers are there that there are many of, and how many are there that there are few of, we actually see that these distributions for each day collapse on top of each other, and so we have a steady state. So the yellow dots here are for the first day of the experiment, and these blue ones are for days 4 to the end of the experiment. And so at some point around day 4, things kind of reach this steady state. And this means that we can do simulations and maybe make distributions like this and compare them. And this is the thing we should be looking at, not so much this. So my colleague Dominique made these simulations, and so I'm plotting here the same sort of idea, which is the distribution of clone sizes for two different expression levels and cell densities. So one of them is for a low cell density and low CRISPR expression, and one of them is for high cell density, high CRISPR expression. And these two are different, although it's not clear what more we can say about it. You can imagine that from the perspective of a bacteria, there are situations where this distribution might matter to it. So for example, at high cell density, there are more large spacer types. So there are more types that have more spacers. And this could be because maybe the bacteria want to keep the good ones around because there are lots of viruses and they want to defend themselves. And maybe at low bacterial density, low CRISPR expression, viruses are not as much of a threat, so they only keep a few spacers, or they keep more options so that they can have more preparedness against unknown threats. So these are the kinds of things we can look for and we can try to say when we're doing this stuff. So just to summarize that last bit there, this is a sort of two-pronged approach to this problem. The one is this mean field description where we can get some qualitatively interesting behavior. And the other is looking at individual bacteria and spacers, and we can get distributions that we can actually compare to the experiment. And I also want to stress that when we're talking about spacers, this is the data that's easiest to get in experiments, because sequencing is much easier than growing things in the lab. So it's much easier to get the spacer data from communities of bacteria. So for example, the Human Microbiome Project, it's much easier to get the data for spacers than to get data for actually like measuring, say, expression versus density. So that's one more reason to do this approach as well. And to summarize the first step I talked about, we can use CRISPR data from a real sample, a real person in a real situation, to make this interaction network, and then we can play around with it and see what it looks like, and maybe hopefully say something about what a healthy microbiome looks like compared to an unhealthy microbiome. Thank you. I would like to acknowledge my group, Sid, my supervisor, Dominique, who did the simulations, and Jem, who's been working a lot on page parser and data analysis. Thank you.
So we have time for questions.
How good is the assumption that the sequences don't change over time? So you're making the assumption that when you take bacteria DNA, and you extract using CRISPR, you try to figure out what its history is.
Yeah.
Is it possible that maybe some sequences change for whatever reason, or yeah?
Yeah, so I think there are two things. So one thing is that the spacers can actually be lost. So it's adding new ones at one end, and other ones can be coming out sort of one way. So definitely there's turnover that way. And are you mean like bacterial mutations, like the bacteria itself could mutate?
Yeah, sure. I mean, I'm not an expert. I'm just thinking about
Yeah.
can it stochastically change or whatever reason?
Right. Yeah. So definitely the bacteria itself could mutate because those repeats are all the same. I'll go back to. Sorry. Yeah, because those because the repeats are all the same, they can be recombined with each other. So you might end up with swapping some orders and using that way too.
Sure. So you probably can't have you maybe can't guarantee a chronological history, but it's at least a record.
Right, and we also don't know when in the past these came around. So you could find that lots of spacers. Yeah, so you might find that, oh, you know, this one, you might think this one's more current, this one is more in the past, but it could have had this sequence for thousands of generations. You don't know when in time it actually got those spacers. So there's that as well. Yeah.
Like eventually wouldn't it be possible to include some like time tracking sequences, like in populations, like you in fact to like have time trackers?
Yeah, so like for example, in this data that I showed, this is exactly sort of the idea. So they're only sequencing the new ones that have been added in this time. And so you can see like, oh, on day one, there were these ones, on day two, there's now also this new one.
Some people have said, like this one, that connects is days and others in generations. Typically, how many generations a day do bacteria?
So it wildly depends depending on the type of bacteria. So the fastest growing ones can divide in about 20 minutes. So that would be like some E. coli. In this data, I think their generation time is about 7 hours. So that's more like 3 generations per day. Yeah.
Would it be possible to influence the CRISPR area, like if you add some DNA and tag it at a particular point of time?
I don't see why not. Yeah, so that's sort of the game that people who are interested in like the food applications are doing. They're trying to give bacteria like good spacers to defeat them, to defeat viruses. So yeah, I think you could do that.
So I guess CRISPR, when the cells divide, or the bacteria divide, they keep the CRISPR community.
Yes.
So don't they over like generations just build up to like 100% to their?
Yeah, so this is, that's a good question. It's not exactly known how this happens. There is a process by which they can be lost, but there also are examples of CRISPR loci that are like huge. So the longest one that I know of is like 580 spacers. But actually, this one, okay. The longest one is over here. But most of them, this is about 3,000 genomes in bacteria. Most of them have only a few spacers. So that could mean that they're being lost or that they're just not being acquired very quickly. Yeah, there's a loss process.
If you sequence one of these CRISPR strands, then what fraction of them will have no match to species that you're trying to match it to? Like, is it 50% or 75 or 10% or 1%?
It's, yeah, many of them don't match. So the other thing is that I can't give you an exact number, but it's sort of dependent on what you're trying to match against. Like a database might or may not have the relevant sequences that you're trying to match against. And the sequences themselves are relatively short. They're about 30 nucleotides long. So you do get matches that are fake basically as well.
Okay. No more questions. Okay, let's add.
Notice that the portions that we do, there are these hairpins that were forward. I'm just curious as to if there's any benefit in forming those hairpins, which is just an enzymatic process, or just trying to minimize the energy.
I don't really know. I imagine that it has some relevance to how it binds with the Cas protein. So this forms some complex, and I think there might be something that the Cas like recognizes this hairpin and binds to. But this, I'm not sure. Yeah.
Okay. All right, so let's thank you again.